Okay, hello. So this is going to be a GCSE Combined Science Trilogy past paper walkthrough for Biology Paper 2, higher tier, and this was the paper that was sat in June 22. Okay, so I'm going to go through and model it how I would have tackled it, and the link to the past paper and the mark scheme is in the description. Okay, so question number 1 then. This question is about the cycling of water and carbon in ecosystems. So it's our water cycle and carbon cycle. Which reaction produces water? So which one is making water? If I go through the options, I've got aerobic, anaerobic and photosynthesis. I should be thinking, right, anaerobic, this is the one that makes lactic acid. So this one doesn't make water. Lactic acid is the only product there. And photosynthesis, well, plants take in water, so it's not going to be that one. It is aerobic, and if you remember the equation, glucose plus oxygen makes CO2 and water. So the water cycle provides water for plants and animals on land before the water goes into lakes and seas. Figure 1 represents the water cycle, and you've got your different arrows here, numbered 1 to 5. What you have to do is name 1 to 5 in these different labels. So I'll annotate it on the actual diagram, but obviously you just put it in the right spaces. So first of all, I've got a lake or a river of water and it's going up into the atmosphere. So 1 would be evaporation. This would then be water vapour, or water as a gas, and then it condenses and forms clouds. So condensation or condenses would be process number 2. Okay, then what happens to the water? Well, it's going to fall down, the clouds precipitate, or precipitation, as rainfall. Now, it might ask you, what are the different types of precipitation? Obviously, you've got rain, but what other forms does water come down from the clouds? Well, it could be hail, or it could be snow. They're all forms of precipitation. As the water then runs down on the earth, it might be absorbed through trees, and it might go up through a tree, taken in by the roots and go out via the leaves. And this is a process you met in Biology Paper 1, which is called transpiration. So number 5 is transpiration. If, however, it doesn't get absorbed by a tree or a plant, it will just run off eventually and go back and find a source of water, a lake or river, etc. And this can be called several things. It can be called draining or drainage, or it can be called runoff, surface runoff. Okay, and that would be accepted as well. So out of those are our 5 marks for our 5 different processes. It then says, in 2007, the population of the world was 6 billion. A study found that 4.5% of the population had a severe water shortage. Calculate how many people had a severe water shortage. So you want 4.5% of 6 billion. So what I would do is do your 6 billion on your calculator. Oh, sorry, one last thing, you've got to give it in standard form. So I would start off, put it in your calculator, 6 billion. What I would then do is find 1%, divide it by 100, times by 4.5 to give me 4.5%, and that gives me an answer of 270 million. So if I get to that point, that would give me 2 marks, but I need to give it in standard form. Remember, standard form is always between 1 and 10, so 1, 2, 3, 4, 5, 6, 7, 8. So my answer would be 2.7 times 10 to the power of 8, and that would be my final answer. Question 1.4. Why do more people have a water shortage now than in 2007? So what is causing a greater water shortage? Is it that climate change has increased the area of deserts? Well, potentially, but we'll look at all of the options, so maybe that one. Each person drinks less water, well, that wouldn't create more water shortage. More water is used to grow crops. Well, yeah, potentially if more water is being used. Sea levels have risen because ice caps are melting. Well, that would have increased the amount of water, so it's not going to be that. And some countries have built desalting factories for seawater. Well, if they're increasing the amount of seawater they're using, that wouldn't affect the water shortage of people. So it's more likely to be the top one and the third one. Okay, question 1.5 then. Leaves on a tree contain carbon compounds. In autumn, the leaves fall to the ground. Microorganisms in the soil recycle carbon so that the leaves, from the leaves, so that carbon is used for new plant growth. Explain how. So it's basically saying you've got leaves on a tree, they die and fall to the ground, and then they're used again for new plant growth. But how does that carbon end back up there? Well, first of all, as your trees fall to the ground in autumn, what happens to them? They decay. So the leaves fall and decay. Okay, now what is actually breaking them down? Well, it's decomposers like bacteria. The bacteria or decomposers respire as they break down the leaves. So you've got the bacteria decomposing and respiring as they're breaking down and decaying the leaves. This releases CO2 into the atmosphere. That would give you your third mark. The final mark is then recognising I've then got CO2 in the atmosphere, but how would it then be used for new plant growth? Well, that CO2 could then be taken in the plants and how they make new plant growth through the process of photosynthesis. This CO2 can then be absorbed for photosynthesis. And that would be your 4 marks there. Question 1.6, what is one benefit of fallen leaves for living plants? Is energy released for living plants? Well, just because leaves have fallen, that's not releasing energy and giving you the plants energy, so that's not it. Insect pests in the soil are killed. No, falling leaves don't kill insects. Nitrates are released into the soil, potentially. Oxygen is supplied to root cells. Well, a dead leaf is not going to give oxygen to a living plant, so it won't be that. So it must be the nitrates. When a plant decomposes, nitrates and other minerals might then be released into the soil and then recycled. And that's the end of question 1. Okay, question 2 then. Water pollution is a problem for humans and wildlife. Explain how human activities are polluting the rivers, lakes and seas. So our sources of fresh water, what are humans doing, or human activities doing to pollute them, and why is that a problem for humans and specifically wildlife? So when you're talking about water pollution, the key process you want to be talking about, you don't need to know this key word, but it's the name of what it's called eutrophication. So my first point would be when sewage or excess fertilisers from farmers run off into water, they cause algae to grow very fast. So if I've got my freshwater supply here, and then I've got loads of chemicals, sewage being dumped into it, the algae plants, they thrive on all that fertiliser or certain types of sewage, and they grow really, really fast on the surface of the water. The algae that is then grown on top of the water then blocks out any sunlight going to below the water. So the algae blocks the sunlight below the surface of the water. Because there's no sunlight, any plants that might be at the bottom of the seabed, these would then die and decompose. Any plants at the bottom of the seabed would then die and decompose. Decompose through decomposers, okay? By decomposers, like bacteria. So all of these plants are then going to be killed and they are then going to die and decompose by bacteria. Now their process, as they are decomposing, the bacteria will be doing respiration, and that respiration uses up any oxygen that was in the water, dissolved into it. The bacteria are going to use it up. So what then happens is the bacteria use up the oxygen in the water through respiration. Now, because then there's no oxygen in the water, any organisms like any fish or any other animals, they would then die because there's no oxygen or limited oxygen in the water. So any fish or animals in water would die due to lack of oxygen. Okay, so therefore we've clearly said how a human activity is affecting our lakes and rivers and specifically affecting wildlife. Okay, and it's caused by two things, sewage and excess fertilisers going into sources of water. This whole process has a fancy scientific name of eutrophication. Okay, you can throw it in if you want, but it's the process and describing it that they're specifically after. Now, other things you can say on this, other points you could make after you describe eutrophication, is more general points really, is that there could be, through human activities, we are increasing toxic chemicals, okay, into water supplies, which are affecting food chains, etc., which build up in food chains. These chemicals can cause mutations in animals, which can obviously harm them. And the other key general point you could say is that what are we doing, especially in the last 30 years or so, we are increasing the amount of plastics in the ocean or in the water, which can affect wildlife because it is not biodegradable. So plastics in the water, which can build up in organisms because it is not biodegradable. Okay, so slightly more general points there, but I think the key one, and allows you to tick a lot of boxes, is the eutrophication answer. And that's just one question here, question 2. Okay, question number 3 then. On a school field, one area of the soil was usually wet, another area of the soil was usually dry. Students investigated the effect of water in the soil on the number of buttercup plants growing in each area. Water is an abiotic factor. Abiotic, not living factor. Name one biotic factor. So this is a living factor that may affect the number of buttercup plants growing on the school field. So again, it's got to be a living factor. So what actually might it be that affects the number of buttercups? Well, I would bring it back to humans. What do we do with fields? We mow them, okay? We either trample or mow the lawns, okay, or the fields. That would be one example. You could also say other living factors might be herbivores coming along and eating the plants. You could say any pathogens or disease coming along, okay? And you could also say competition. Potentially there are different plants that are better adapted and they're not allowing the buttercups to plant. These are all examples of biotic factors. Question 3.2. Describe a method to investigate if the amount of water in the soil affects the number of buttercups in the field. Okay, so this is a design question, and hopefully you remember this is your quadrat measuring species abundance required practical. So how do I go about estimating or seeing whether buttercups grow better in wet soil or dry soil? So the first thing I would do is measure an area of wet soil and dry soil. Now it's your plan, so you can make it up. You might just say 100 metres squared. Okay, obviously quite a big area, but it's your plan. You can put what you want, any specific measurements. So you measure an area. What you would then do is we get your quadrat. So get quadrat. And you need to place it onto your wet and dry areas. But how do you know where to place it? We never just chuck it, okay, and just hope. We've always got to make sure it's totally random. So get quadrat and we place it randomly. And I would suggest we're using a random number generator. Okay, so you could get your field, divide it up into loads of grids, give each of them a number, and then you would put your quadrat down in whatever number grid it says, and this whole thing reduces bias. So you get your quadrat, you place it randomly. You would then count the number of buttercup plants in the quadrat, okay, and you would do this times 10 in each area. Okay, so you do it 10 times in the wet area, 10 times in the dry area. You would then calculate a mean per metre squared for wet and dry soil. So you get a mean number of buttercups per metre squared in the wet soil and a mean number per the dry soil. You would then times the mean per metre squared by the area to give you an estimate of the population in that area. Okay, now the only other thing you could then do is, because it's specifically, and you could say compare populations or compare estimates for wet and dry soil. You could, if you want to add a bit more data to it, use an actual moisture sensor. Okay, so you could specifically at this point, you could also add in take moisture readings. Okay, there are certain devices, moisture sensors, that just tell you how much water there is in the soil, and that would allow you to do potentially, instead of just saying wet and dry, you can say exactly how wet, how dry. But generally this is your standard quadrat method that you can apply for any organism on a field or specific habitat. And that's the end of question 3. Okay, question number 4 then. The scientists investigated the effect of soil nitrate ion concentration on the yield of corn. This is the method they used. Corn plants were grown in a large box of soil. The soil nitrate ion concentration in a box was kept at 0 parts per million, ppm. All the corn from each plant in the box was removed and weighed. The mean mass of the corn plant was calculated. Steps 1 to 4 were repeated for boxes containing soil with different concentration of ammonium nitrates. So we've done it for increasing amounts, and figure 2 shows corn plants in the box with a 20 parts per million soil nitrate ion concentration. Give 2 variables the students should have controlled, kept the same in this investigation. So they're changing, their independent variable is their concentration of nitrate ions. Their dependent variable is the mass of corn that was being produced. So what should they have kept the same? You use the diagram as well. So you've got corn plants growing in a box, so there's loads of different things you can say here. What other factors might affect how well a plant grows? So for the sake of time, there's loads you can put here. So I'm just going to say amount of light the corn plants might have, amount of water, are they growing at the same temperature? Do they have the same amount of soil? Do they have the same soil type or pH? Is there the same number of plants growing in the box? Is it the same type of corn? And were they allowed exactly the same amount of time before harvesting? You can't just say time, but you have to say the time before it was harvested. Okay, so loads of different control variables, any 2 of them would get you 2 marks there. The scientists then carried out a valid investigation. Table 1 shows the scientists' results. So you've got the soil nitrate ion concentration in parts per million, and you've got the mean mass of corn per plant in grams. So what we then have to do is do a graph. We need to label the y-axis, use a suitable scale for the y-axis, plot the data, draw a line of best fit. So tick them off as we go, label the y-axis. So if it's soil nitrate ion concentration is at the bottom, our other one must be on our y-axis. So mean mass of corn per plant in grams. So just copy that out, mean mass of plant corn in grams. And then we need to decide on a scale for our y-axis. So I can see here, I've got to be able to get up to 268 if I'm going to be able to put it onto my graph. So I then need to think of a scale that will use the majority of the graph paper. Okay, I've got to get up to 268 or 270. So in order to do that, if I did it in 50s, 50, 100, 100, 50, 200, 250, 300, I mean, yeah, you might just get away with doing that as an appropriate scale, but you're missing out a large proportion, or 3 big squares off the graph paper. You want to try and make it as big as possible. You also want to choose a relatively easy number for you to plot your data. So I would go up in 40s. So each half square would be 20. So 40, 80, 120, 160, 200, 240, 280. And I can see here, I've pretty much used most of my graph paper. What I'm then going to do is start plotting on the data. So I've done the y-axis, I've then used a suitable scale. I need to plot the data from table 1, just take it one at a time. Remember, you've always got to be within half a square. Okay, so for the sake of time, to save you having to watch me scroll back and forward, I've plotted the points on, been really accurate, making sure it's within half a square each for my plotting. If you don't get it within half a square, you won't get the marks. You've then got to do the final line of best fit. So what you have to do is obviously a curve. It would go up, turn the paper to help you then do it, and then it would curve back down. Majority of the time in science, it would be a curved line of best fit. and that is your final graph. So we have label the y-axis, use a suitable scale, plotted the data, draw the line of best fit clearly on it. One smooth line in pencil, no extra shading, and that is your graph done. Table 1 then is then repeated below. Okay, you've got the soil nitrate ion concentration and the mean mass as before. Describe the relationship in soil nitrate ion concentration and the mean mass of corn per plant. Use data from table 1 in your answer. So describe just means say what you see. So what's happening to the numbers as I increase my concentration of nitrate ion? So I would say as the concentration of nitrate ions increase, I can see that it's going up. Okay, the mean mass of corn, use the labels in the table, corn increases. But it doesn't always keep going up. It goes up initially up to 30 parts per million. Okay, with a mass of 268 grams. However, after 30, what is happening? Our mass is then decreasing as I add more nitrate ions. Above 30 ppm, the mass of the corn decreases. Now, in order to get the marks here, you need to get 1 mark for saying it increases and you have to say it decreases, and 1 mark for identifying that the mean mass, the maximum mass is at 30 with 268. So make sure you always talk about the peak or the highest amount as well on the table. Question 4.4. Farmers add nitrate fertiliser to fields where they grow corn, but nitrate fertilisers are expensive. Evaluate the economic and environmental implications of adding fertiliser to soil in nitrate ion concentrations from 0 to 50. So evaluate, so saying the effects, positive and negative, of economic, so that's money, and environmental implications of adding fertiliser to soil. So it's basically explaining the trends that you've just identified. So I would be thinking, right, economic, if I'm going to add more fertiliser, I want more crops. That works between 0 and 30. So I would say increasing the nitrate ion concentration increases the mass of crops and therefore the profit for farmers. Okay, up to 30 parts per million. Okay, because we know from the table above, 30 is our peak. Okay, however, if we go above 30, we know it goes down, so we can say that as well. Adding fertiliser above 30 parts per million would decrease the yield and reduce the profit. Okay, so if you're adding too much and you're spending loads of fertiliser but you're not getting as much crop, that's going to hit your profit margins. So what I would then say is this wastes money. The next thing I could say is therefore adding the right amount to the soil must balance growth of plants and cost of fertiliser. Okay, so it's a balance as to how much you add. And how much you add would actually depend on how much the starting amount of fertiliser in the soil as well. But I also need to link in environmental implications. Well, what's the issue of adding too much fertiliser? Adding a lot of fertiliser to fields can cause runoff. It can cause runoff into water sources, which can cause pollution. Now, you don't need to rewrite about eutrophication on this answer because it's you've already got enough points. It's not asking you to explain that. That was on the previous question. But it's just saying, generally, obviously adding too much fertiliser would then cause environmental impacts again. Okay, and that's the end of question 4. Okay, question number 5 then. Blood glucose concentration in the human body needs to be kept within the normal range. Figure 4 shows that 2 hormones control blood glucose concentration. Name the type of hormonal control shown in figure 4. So we can see here, we've got our blood glucose concentration, and when it's too low, it goes to the pancreas, and hormone A is released, and it brings it back to normal. When it's too high, information is sent to the pancreas, which then releases hormone B, which then reduces it or brings it back down to normal. So what is this process called when, if something gets too high, your body works to bring it back down to normal? Now, in this mark scheme, it is called, and you have to put negative feedback. Okay, so if something goes up, your body will work to bring it back down. It's an example of negative feedback. It doesn't accept homeostasis as an answer in that one. Name hormones A and B in figure 4. So if I look in hormone A, what is hormone A doing to our blood glucose concentration? Well, hormone A is taking it and it's bringing it back to normal from when it was too low. So hormone A is the hormone when our blood sugar levels are too low, and that is for one, it's the higher tier only hormone you've got to know, which is called glucagon. Now, blood glucose concentration when it gets too high, this is the classic one that everyone's got to know, which is insulin. Okay, so glucagon is A, insulin is B. You then got a classic 6 marker. Explain how 2 hormones in figure 4 keep the blood glucose concentration within the normal range for 3 hours after a meal. So it's basically, how does your body regulate your blood glucose levels? So there's lots you can say here in order to get the marks. So I would say, after a meal, the blood glucose levels will increase. Okay, in order to then counteract that and bring it back down, the pancreas will release insulin. The pancreas releases insulin to reduce the blood glucose levels. And what effect does that glucose then have? The glucose causes cells to increase the uptake of glucose from the blood. So therefore our cells are using it quicker, bringing it back down. And then it also causes the liver to convert glucose to glycogen. So glycogen is just the method that our liver stores glucose to bring back down the blood sugar levels. So that's what happens if it gets too high. And then we can then describe what happens when it gets too low. When blood sugar levels are too low, the pancreas releases our other hormone, glucagon. Sorry if I can type. Releases glucagon. Glucagon causes the liver to convert the stored glycogen back to glucose. This increases the blood glucose concentration. Okay, so you've break your answer down clearly. What happens when your blood sugar levels get too low, or get too high, sorry, and then what happens when they get too low. Okay, plenty you can put into that one. Probably a couple more than 6 marks there, but loads of information you can put in. Then, moving on to 5.4. Female reproductive hormones are used to treat fertility in women. Follicle-stimulating hormone and then luteinizing hormone can be injected into a woman to help her become pregnant by sexual intercourse. Explain how injecting FSH and LH will help a woman become pregnant. So this is basically knowing what are the jobs of FSH and LH. Well, FSH, follicle stimulating hormone, the job of it is to mature the egg. FSH matures eggs in the woman's ovaries. LH then causes ovulation, which releases the eggs. Okay, so if I've got more eggs being matured and more eggs being released, so therefore there is a greater chance of a mature egg being fertilized. Okay, and you've got to say the job for each one and the impact on it to get the 3 marks. Okay, and that is 5.4. 5.5 then, in some women, the injections of FSH and LH are the first steps in IVF, in vitro fertilization. Describe the remaining steps in IVF. So what happens is you get the mature eggs, okay, and they've been released. So what then happens in IVF after that? Well, IVF is in vitro fertilization, which is artificial fertilization out of the woman's body. So the mature eggs are collected and harvested. Then scientists would artificially put the sperm in. They are fertilized in a laboratory. What then happens is those mature eggs hopefully will develop into an embryo. The eggs, or fertilized eggs, develop into an embryo. Then, after that it's an embryo, it can then be implanted back into the uterus of the woman. The embryo can be inserted or placed into the womb or uterus of the mother. And then hopefully it will go on to develop into a baby. Now, in terms of IVF then, you've got to know that it's fertilised obviously outside of the body, and the success rates obviously of it are quite low because, first of all, whilst it may become fertilised, it might not develop into an embryo, and then the success rates of then getting those embryos and successfully implanting them into the mother are actually quite low. Okay, so IVF, you get some free rounds on the NHS, but then you also have to be in good health and having a healthy lifestyle in order for it to be approved. Question 5.6. There are 2 different processes of cell division in humans. Describe 3 differences between cell division to form sperm cells compared with cell division to form liver cells. So, I should be thinking, what are my 2 types of cell division? Well, I've got mitosis, and because this is paper 2, I've also got to know about meiosis as well. So, sperm cells are produced by meiosis, whereas liver cells are mitosis. So that would be my first comparison, 3 differences. So I say liver cells are produced by mitosis, whereas sperm cells are produced by meiosis. And then I can think of some of my differences between mitosis and meiosis. What's different about it? Well, in mitosis, in liver cells, there is 1 division. And 2 daughter cells. Whereas in sperms, which is meiosis, there are 2 divisions and 4 daughter cells. Another difference that I can then say, as for my third one, is actually what's different about a sperm cell compared to a liver cell. Well, sperm cells are gametes that have half the amount of DNA. So sperm cells, or from meiosis, have half the amount of DNA, which is 23 chromosomes, whereas liver cells have 46 chromosomes. The other difference that you can then say is that sperm cells, the process of meiosis, gives variation. Okay, all the sperm cells are different, but mitosis is genetically identical. So a few more comparisons than you actually need there, but any of them would then get you those 3 marks. And that's a big question, 18 marks, and that's the end of question 5. Okay, question number 6 then. The polar bear is a mammal that lives in Arctic conditions. Complete table 2 for the classification of the polar bear, and that's its binomial name. Okay, so this is when you've got to know your classification systems. Now they've combined here Carl Woos's classification with his 3 domains with the classic Carl Linnaeus system going down from kingdom to species. So remember, Carl Woos's 3 domains sit above the kingdoms. So the domains of the Archaea, Eukaryota, and Prokaryota. Sometimes teachers aren't clear in establishing that the domains can sit above the kingdoms, whereas the traditional way of doing it is the Carl Linnaeus system. So we've got an animal, a polar bear. What domain would the polar bear come under? Well, it would be the Eukaryota, okay, domain system. And then what kingdom would a polar bear come in? Well, it is an animal, so it would just be the animal kingdom. Then what two things are missing from Cole Linnaeus' system? You've got kingdom, then it should be phylum, that's missing, class, order, then family, genus, and then species. Okay, you've just got to know the order of Cole Linnaeus' system, and remember that Cole's three-domain system sits above it. Scientists have been measuring the area of sea ice in the Arctic since 1980. Figure 5 shows the area covered by sea ice every September. So you've got the year and the area of sea ice in millions of square kilometres. Determine the annual rate of sea ice between 1985 and 2017. A trend line has been drawn for you. So we are working out the rate of loss per million square kilometres per year. So how much ice is being lost per year between 1985 and 2017, okay, which would be at this point here on my graph. Now you've already got the line of best fit given to you, so you make sure you will use that line of best fit. So for 1985, drawing along to our line of best fit and along, you are allowed any number between 7.7 and 7.8. You go in the middle, 7.75, that would be totally fine as well. Okay, so any of those numbers for 1985, and for 2017, you are allowed any number going along, okay, trying to be as accurate as possible. Obviously, you with a pencil and ruler would be much more accurate, but you would be allowed any number between 4.3 and 4.4. You can kind of see this, where they've put it, it's just above 4.3. So you might say 4.32, but again, any value between those two would be accepted. So what I can then do, let's say I'm going to use 7.8, okay, and I'm going to use 4.3. Okay, if I want to then know how much sea ice have I lost over this period of time, well, I would just do 7.8 minus 4.3, do that on my calculator, and then that would give me an answer. 7.8 minus 4.3 would give me an answer of 3.5. So that's how much ice has been lost. But I want to know how much ice has been lost per year, so I would just divide it by how many years there's been. If I work out how many years are between 1985 and 2017, well, that would give me 32 years. So I would do 3.5 divided by 32 to give me an answer of 0.109375. Okay, it hasn't said anything about significant figures, so I can just leave it like that. Now everyone's answers will be slightly different depending on what numbers you actually picked from the ranges here, but if you picked any two numbers within those two ranges I said, you would get 3 marks on it. Okay, right, the next question then. The total number of polar bears living in the Arctic ice is not known. The hunting of polar bears has been banned or reduced in some areas. In some populations, the average mass and height of polar bears has decreased. So the mass and height of them has decreased. Polar bears eat seals. Seals live on the sea ice in winter and raise their pups there in early spring. In the summer, seals live mainly in the sea, catching fish to eat. Polar bears spend much of the year hunting seals on sea ice and in the sea nearby. The sea ice area is at its lowest each year in September at the end of summer. The polar bears feed mainly in early spring and again in autumn to build up fat stores to survive the next winter. During the winter of 2017, scientists measured the metabolic rate of 9 female polar bears and found them to be much higher than expected. So they've got high metabolic rates. Cameras attached to the female polar bears showed that they had to swim long distances to find seals to eat. Suggests why polar bears find it harder to catch seals in the autumn than the spring. Okay, so we just need to use this information here to help us break it down. So why is it harder for them to catch seals in the autumn rather than the spring? Okay, well, what are the seals doing in the spring? It says here, in the summer, seals live mainly in the sea, catching fish to eat. So this is quite a hard question. We've given you lots of information, and it's all about trying to pull out the key points. So what I would say is most seals are in the sea in autumn, okay, therefore harder to catch. Okay, then why else could it be? Well, what is actually happening in autumn? Well, in autumn, there could just be more polar bears about, so there might be more competition. But also, if it's later on in the year, okay, what's happened to the seals? The seals have then grown through the summer rather than the spring. Because it says seals live on the ice in winter and raise their pups there in winter, so therefore come the autumn they'll be bigger. The seals would also be bigger and harder to catch. And that would be the answer to 6.3. Okay, question 6.4 then, last question on this paper. Evaluate what might happen to the population of polar bears in the Arctic in the future. So what do we think or know what's going to happen to them? Well, I think the logical thing is that the amount of polar bears, you've been told, the mass and height of them are decreasing. So why might that be happening? Well, we know that actually there's much less sea ice because polar ice caps are melting. And therefore, what does that mean? Well, in terms of seals, the seals would then have less ice to then live on, and therefore polar bears would then find it much harder to hunt the seals. So I'd be saying global warming or climate change is melting sea ice. This reduces the habitat of polar bears and seals. Okay, therefore there might be less seals to hunt and eat. Now the issue that they've also got to find, if they've got a decreasing number of seals, they're going to have to travel further between different pieces of sea ice. They may also have to swim further to catch more seals. Okay, using more energy. Now, if they're using more energy then to swim and catch more seals, this would explain why their mass is then decreasing. You could then also say they would not build up enough fat reserves for the winter, where they wouldn't be hunting as much. Now, because it's evaluate, these are reasons why it may decrease. You could also then say reasons why it could potentially increase. Well, if we go back to the very beginning of the explanation, it mentions about polar bears the hunting of polar bears has been banned or reduced in some areas. So you could also then say, as a reason it might increase, is that they could increase if hunting is totally banned or actually enforced, and therefore you could also say it would increase if we then stop climate change. So just quickly, increase, they might increase if we expand or enforce hunting and successfully reduce climate change. But obviously the more logical or more, not logical, more likely reason is that they're more likely they're going to be decreasing in number. Okay, and that brings us to the end of the paper. Right, I hope it's been useful, and remember the paper and the mark scheme are in the description. Thank you.